Hello biology students, and welcome to video 3 of the DNA technology unit. So far in the first 2 videos in this unit, we talked about the cloning process by which we create recombinant DNA and then put that recombinant DNA into a different host organism, usually a bacteria, to let that bacteria produce human protein for us. So we've been on this first use for the entirety of videos 1 and 2. So now that we're in video 3, we're going to look at how we can use DNA technology to identify people. In other words, how we can use DNA fingerprinting to solve crimes, identify who's the father in a paternity case, and things like that. So here we are. So use number 2, which is identifying people, and we're going to use the example of forensic analysis or DNA profiling to look at how these techniques work. So there are 3 main steps that we have to go through if we're going to use DNA in order to identify a person. So the first thing we have to do is isolate that DNA and make lots of copies of it using a process called PCR that we'll look at in a second. And basically, this would look like if you were at a crime scene, you might find the DNA from a criminal and then make lots of copies of that DNA so that you would have plenty of DNA to use to analyze. The next thing we're going to do is sort the DNA fragments containing STRs by gel electrophoresis. So that's a lot of big words that we haven't seen yet, but basically the idea is here is that we're going to look at unique sections of DNA called STRs that are different for each person, and then we're going to use a technique called gel electrophoresis to separate those unique regions by size so we can analyze them. And then finally, we're going to actually do the analysis. We're going to compare these unique regions, these STRs, to try to identify who the DNA belongs to. Okay, so let's look at the first step first. So the first step we said was to isolate the DNA and make many copies of it. So this is important because sometimes samples of DNA are too scarce or impure to use. So, on a crime scene, you might find a couple of skin cells or 1 drop of blood, and that's not enough DNA to be able to reliably analyze it. So therefore, one of the first things scientists have to do, and in fact, you do this whenever you want to make a lot of DNA, not just for crime scene analysis, is you can use this reaction called PCR, which stands for polymerase chain reaction, and that allows you to make billions of copies of one piece of DNA in just a couple of hours, okay? So one thing I want to point out is you're just making a lot of copies of a small section. You're not copying, so maybe if I found a cell that had an entire 46 chromosomes from a person, I wouldn't make billions of copies of all that DNA. That would take too long, and I don't need all that DNA. I'm just going to make copies of one section. And if you remember from the previous slide, I talked about those STRs. STRs are regions of the DNA that are unique to a person. So if I made lots of copies of those unique regions, just those STR regions, I would now have plenty of DNA that I could analyze. Okay, so now let's look at the steps of PCR. So basically, PCR is a machine. A PCR machine is a machine that basically changes the temperature of the solution that DNA is in cycles. Okay, so the first step of one cycle would be to heat up the DNA to 95 degrees Celsius. And the reason we do this is because we want to denature the DNA. If I have my DNA like this, I want to separate the strands so they're separated. Okay. So you can kind of think the enzyme helicase did this in DNA replication, but we don't use helicase, we just use heat. Because the hydrogen bonds are so weak compared to the covalent bonds that make up the backbone, if you heat it up nice and high, those bonds will break and the strands will separate. So the next step is the annealing step, and basically what we do is we add a little primer, and a primer is a little piece of DNA that's complementary to my DNA. So I add a little primer that's complementary to the 2 ends, and that acts as a starting point for DNA polymerase to start replicating my DNA. So I give it a starting point, that primer. So the primers are complementary to the one primer is complementary to one end of the DNA, and the other primer is complementary to the other end of the other strand. So I have one primer that will attach here and one primer that will attach here. Okay? And then this happens at between 50 and 65 degrees Celsius because it has to be cool enough that these primers, these little sequences of DNA, can hydrogen bond to the single-stranded DNA. Okay. So at this point, I have 2 strands of DNA, and both of them I've started replicating. So I've added a little primer. Okay. Then finally, the third step is at 72 degrees Celsius, so pretty warm. There's going to be some DNA polymerase in my reaction already. It's something I would have added to the test tube before I put it in the machine. Now the machine's going to go to 72 degrees Celsius, and this is the perfect temperature for DNA polymerase. So in my reaction test tube, there's going to be my DNA, there's going to be DNA polymerase, and there's also going to be free nucleotides, so extra A's, extra T's, extra C's, and extra G's. And the DNA polymerase is going to, starting at the primer, it's going to elongate my DNA, so now I have 2 pieces of DNA, okay? So now DNA polymerase, you would think, because the DNA polymerase has been in this test tube this entire time, so you think way back when I went up to 95 degrees Celsius, you would think the DNA polymerase would denature, but it does not, because we use special DNA polymerase that comes from bacteria that live in hot springs. So that their DNA polymerase has evolved to withstand high temperatures. Okay, so turns out PCR, 1 cycle takes like 45 seconds. It takes 45 seconds to go to 95 degrees, to 50 degrees, and then 72. So this can happen fairly quickly. And if you do between 20 and 35 cycles, so about a half an hour, you can exponentially amplify your DNA very, very quickly. So after 1 minute, after 1 cycle, if you start with 1 piece of DNA, after 1 cycle you'll have 2. And then after the second round, those 2 will become 4. And then those 4 will become 8, and by the time you get to the 35th round, you're going to have 34 billion molecules. So if each round takes about a minute, you only need about 35 minutes to go from 1 piece of DNA to 34 billion, which is quite a lot. Okay, so you can kind of see how that would work here. After my first round, I would have 2 pieces, and then those 2 would amplify to 4 in the next round, etc., until after 35 rounds, we had 34 billion pieces of DNA. Okay, so if I am using PCR to do DNA fingerprinting, to identify a criminal or something like that, then I would, instead of just amplifying any old section of the DNA, instead I would choose to use PCR to amplify STR regions. STR stands for short tandem repeat, and as I talked about earlier in the video, these are regions of DNA that are basically it's 4 nucleotides repeated many, many times. So maybe it's AAGG repeated over and over again. So it's AAGG, AAGG, AAGG. And the number of repeats in a given person varies wildly. So maybe I only have 20 repeats of this AAGG, but one of you might have 25 repeats. Okay? So the idea is we take 13 of these STRs, so 13 different repeated sections, and we use PCR to amplify all of them. Okay? And my 13 sections are all going to be different lengths than your 13 sections. And then what we do is we use this technique called gel electrophoresis to separate these PCR'd STR fragments by size. So the way a gel electrophoresis works, it's literally a gel. It's kind of like a thick jelly. And it's set up so it has electricity running through it. And I add DNA to my gel, and I dye it so I can see it. And when I turn on the electricity, the DNA starts moving through the gel. And if a piece is very big, it can't move through the gel very well. It's too big, it's bulky. The gel has little microscopic holes, but if a really big piece of DNA, it's not going to travel through the holes very easily, so it's going to move slowly. And then my small pieces of DNA are going to easily move through the holes in the gel, so they're going to move very quickly. Okay? And you might be wondering, well, why does DNA move through the gel when you turn electricity on? And the reason for that is DNA, remember, is negatively charged because the phosphate group of DNA is negatively charged. So if I set up my gel, well, here's my little gel, if I have a positive end of my electrical current down here and a negative end up here, and I add my DNA to be right here, when I turn on the electricity, the DNA is going to want to move towards the positive and away from the negative. Okay, so here's what a gel looks like. So you can see I put in my DNA right at the top, Here's my negative electrode, and here's my positive electrode. If I turn on the power source, the DNA is going to start moving through the gel, and the smallest fragments are going to move most quickly. So you can see these ones down here are smaller than these ones up here. And then when the gel is done running, you turn it off, and you can compare. So maybe here I have the DNA from suspect 1, here I have the DNA from maybe suspect 2, and maybe here I have the DNA from the crime scene. So if we look, this first piece of DNA right here, maybe that's the first STR we're analyzing. And it looks like, if I look closely, it looks like they're about the same. So then maybe that STR wasn't useful for us. But maybe we can look at the second STR that I amplify with PCR, this one right here. Okay. And if we look, it looks like this one right here is the same, the one right here is the same size as the one right here, and this one right here is a little bit smaller. So that suggests that suspect 1, their DNA matches the DNA at the crime scene. And you can do this for each STR. You can compare and see if you have a match. Okay, so this is a picture of an actual gel. This is what it actually looks like. You can make them glow. You can use a dye that's fluorescent, and then you can compare, you know, the DNA from your suspects to the DNA found at the crime scene and see if they have matching STRs. Are their STRs the same size? Turns out this is very, very reliable because, like I said, we use 13 different regions of DNA, and the probability that 2 people will match at one region is 1 in 10. But if you use 13 different regions, 1 in 10 times 1 in 10 times 1 in 10, you know, times 13 times, means your probability of having an identical 13 regions to somebody else is virtually 0. So this is a very reliable method to identify someone using their DNA. So that's it for video 3, and I'll see you in video 4.